So I too would like to thank the organizing committee, which I was part of, but especially for the leadership that we had from Barbara Schneemann. I've been tasked with addressing sort of the underlying biological process for special nutritional requirements. And so I'll be speaking to this. Again, this is an area that is very much in its infancy, but I think one that's very important. These are my disclosures. And so first I'd like to start with this issue of science back in 2010 that asked the question, can science feed the world? And this dealt with this concern that within a few decades, there's going to be 9 to 10 billion people on this planet. And are we going to be able to feed them? And I always find it ironic here that on that same cover is an issue of diabetes. And really the question is, not can we feed the world, because I think we can, and production agriculture certainly, it's believed, has the capacity to do this, but can we meet the nutrient needs of all the people that are going to be living on this planet? And that's the big question, because we do have an unprecedented capacity to really develop all sorts of foods. And the question, how can we match those foods and those nutrients to really support all human health? And so that really leads to the question that we've been asking for years, is related to our dietary and nutrient recommendations, and that is adequate for what? And as Dr. Brannon talked to a few minutes ago, there really has been an evolution where we used to talk about avoiding deficiency, then really talking about metabolic function, and then more recently chronic disease prevention, and today we're going to be talking about disease management. So there's a lot of activity in this area, especially the last couple of years, and Dr. Schneemann referred to some of this, but recently the National Academy was tasked with developing a redesign process for the dietary guidelines. And within that task was the question, how the DGA can better prevent chronic disease, ensure nutritional sufficiency for all Americans, and accommodate a range of individual factors. And those individual factors include age, gender, and metabolic health. So clearly we're talking about how food can maintain health in people who already have challenged metabolic health. And of course, there was also the report that was released later last year on the development of a framework for inserting chronic disease endpoints in the derivation of our dietary reference intakes. And I think although this is about really prevention of chronic disease and using nutrient requirements to prevent, many of the aspects of that report are highly relevant to the discussion today. And that is, number one, chronic diseases are complex traits. One of the biggest risk factors for chronic disease is age. But we know that risk for chronic disease is an interaction between age and diet and genetics and epigenetics, other environmental factors. So these are truly complex traits for which diet is just one component. Number two, few chronic diseases are affected by a single nutrient or a single pathway. And so therefore, we really have to consider systems and networks over pathways when we think about what dietary requirements are in the context of both disease prevention as well as managing disease. So we have to consider, again, systems over pathways. And it really requires us to rethink biomarkers, because what we need are integrative biomarkers that really capture a system and the interaction of all the nutrients within that system and all of the other inherent biological factors like genetic variation. We need to understand biomarkers of aging. Again, if aging is a risk for chronic disease, we have to understand what are the biomarkers of aging, and then how does nutrition or nutrient supply alter that trajectory of aging, whether or not you're moving towards healthy aging or moving towards chronic disease. And importantly, the committee really established that any recommendation coming out of a chronic disease endpoint really should have a moderate level of evidence using grade standards. That's a very high bar for the nutrition community. And in fact, when you look at the relationship between any nutrient and a chronic disease, for very, very few do we have a moderate level of evidence to support a recommendation. So again, I would posit that when we think about now special nutritional requirements, all of these are relevant. Everything that was relevant to chronic disease prevention would really hold for how we think about management of chronic disease. And so here, when we think about, nutrient needs and our dietary reference intakes, we know again that the amount of nutrients that we need are determined by a number of physiological processes, including absorption, catabolism of those nutrients, excretion, metabolism, stability, transport, bioactivation, the energetic state, nutrient storage. All of these factors can influence how much of a given nutrient we need. And it is variations in these processes that give us the variation among individuals in terms of their optimal level of nutrient intake. But we also know for all of these biological processes, there are modifiers and sensitizers. And I'll start from the bottom. We know that sex, pregnancy, lactation, age, all influence our nutrient requirements. And in fact, often for DRIs, we have subgroups in these categories that have a different level of requirement than others. But we now know there are other factors: microbiome, pharmaceuticals, toxins, nutrient-nutrient interactions, genetics, food matrix, epigenetics, all can modify these processes and give us variation and requirement in the population. But we also know, and that's what we're going to talk about today, that disease can be a major modifier of these processes. And in fact, there's evidence can then alter what our nutrient requirements are. So this slide here really tries to capture where we are in terms of thinking through what nutrient requirements are for different populations. We can think about nutrient requirements required for health. That is, in healthy population, what are the level of intake that's required to maintain whole body nutritional status, normal physiological function, clinical outcomes in terms of prevention of chronic disease, and those predictive biomarkers we need to know that tell us what we eat today, how that's going to affect our risk of chronic disease tomorrow or its onset. And clearly, the DRIs now are really capturing this health space and into this disease prevention, both primary and secondary disease prevention space. But recent estimates are that about half of the US population suffers from a chronic disease and is being treated for a chronic disease. And so this is a problem for us when, in theory, the DRIs may not hold for 50% of the US population if you believe that chronic disease modifies your optimal nutrient intakes or your requirement. So really now what we're talking about today is dealing with this other population that has chronic disease, may be treated for chronic disease, and how do we think about what their nutrient requirements are. And when we think about it, we still need to understand all of these outcomes. That is, what is the effect of disease on whole body nutritional status, on normal physiological function, on clinical outcomes, and understanding these predictive biomarkers. But we have other considerations here. We need to think about comorbidities. And that is, when we think about special nutritional requirements, we're not talking about nutrition in any way curing or mitigating that disease. What we're talking about is how the nutritional requirements are changed because of the disease. But we can think about comorbidities. There is evidence that some of the comorbidities associated with a disease are due to the effect of disease on the nutritional status or nutritional function that then make you susceptible to other diseases. So you might think about diabetes and diabetic neuropathy, for example. That's a comorbidity. Is diabetic neuropathy due to some sort of a nutrient deficiency related to the diabetes? We need to think about tissue-specific nutritional status, as Dr. Brandon mentioned. That is, sometimes disease is isolated to a single tissue. The single tissue is diseased. You may not have a systemic effect. How do you go then and measure and quantify what the nutritional needs are of that diseased tissue when you may not have whole body indicators of the effect of a nutritional deficiency or a nutritional excess? We can talk about restoration of function. That is, if disease causes a functional deterioration in metabolism or physiology, can we provide new inputs to restore some of that function that's been lost because of disease? And we have to talk about tissue regeneration. So often in disease, we go through a period where we need to renew that tissue. And then we have to begin to think about stem cells and what are the special nutritional requirements that really optimize the number of stem cells we have, the quality of those stem cells, and their ability to, again, restore that organ, both in terms of physically the number of cells, but also their function. And so that's really what we want to focus on in terms of special nutritional requirements, is what are these new measures and new biology that we really need to understand. So when we think about special nutritional requirements, we have to think about adequacy. Adequacy for whom? That is, are we talking about clinical populations, or are we talking about individuals? That is, can we take a population approach? Can we classify chronic disease in such a way that it becomes a public health issue and not an issue of individuals and N equals 1? How much heterogeneity is there in the population that you can classify? We have to ask the question, adequate for what? And we actually have a better handle on that, because really what we're trying to address is a disease-induced change in nutritional needs. That's what we need to address. Again, we have to be careful that we really think about what is the difference between a special nutritional requirement and what is a drug. Drugs cure, mitigate, or treat a primary disease, and that's not what we're talking about here. We're talking about how, again, a disease causes a nutritional deficiency. We do have to keep in mind, though, that many people who have chronic disease are being treated for that chronic disease, and drugs can alter nutrient requirements independent of the disease, and then that change in nutritional requirements could lead to a secondary disease if those requirements are not met. We also have to think about safety, and do we think about this in terms of a clinical or a public health paradigm? And that is, if someone has a special nutritional requirement, they probably are being seen by a physician. And so what does safety mean in a public health sense? What does it mean for someone who's under clinical care? And are those paradigms the same or not? But these are, I think, some of the questions we have to think about. In terms of drugs, again, we have to keep in mind that people with chronic disease are taking drugs, and drugs can alter nutrition needs. This was a commentary that was published a couple years ago, and it really was a commentary on this trial that was done in China looking at enalapril, which is an antihypertensive that was investigated in terms of its effect on renal disease. And what this found was that the drug alone actually altered serum folate levels. And serum folate levels are an indicator of nutritional adequacy. So the drug had one effect on actually altering serum levels. Whether this is meaningful in terms of folate requirements or folic acid requirements isn't clear, but it was also shown that folic acid given in conjunction with the drug was much more efficacious in terms of the outcome of renal disease. So drugs can alter nutrition needs, they can alter physiology, they can alter biomarkers of nutritional status, and they can affect efficacy of different drugs. So we have to think about that in terms of clinical populations. And in fact, very, very recently, there was a review that was just published that looked at a variety of drugs and their effects on nutrient levels and biomarkers of nutrient levels. So this is really something that really goes hand in glove with chronic disease, that is the effect of drugs on nutrition as well. Again, special nutrition needs are not treating a primary disease. So one of the questions we have to ask is the nutrient intervention having a physiological or drug, meaning an off-target effect? So I just offered this one definition, that special nutrition needs act through evolutionarily derived physiological mechanisms as opposed to off-target effects to restore nutritional adequacy and physiological function and thereby manage a specific disease state. So this is in contrast to this example here, which might be an example where a nutrient is acting like a drug. And that is pharmacological doses, that is 10x the DRI of niacin, or actually the RDA, is given to improve lipid profiles. Niacin works, nicotinamide does not. Evidence is that these patients, this population, doesn't have a niacin deficiency. But if you give them a lot of niacin, you improve lipid profiles. So this probably would not be an example of a special nutritional need or requirement. This is a case where you have a nutrient that's having some sort of an off-target effect that we don't understand, that's showing clinical improvement, but it's not acting as a nutrient, it's acting more as a drug. So when we think about how we consider proposed standards for special nutritional needs, we first have to establish a robust biological premise. That is, how and why are the nutritional needs different? And what are the relevant biomarkers of these altered nutrient needs? Can we use the same biomarkers that we're using for healthy populations? And I'll talk about that later. We have to ask the question of efficacy. Does the increased or decreased intake address disease-induced changes in nutrient needs? Does it affect nutritional status and or nutrient function? And are these effects chronic or are they acute? And then we have to ask the question, does the altered intake improve a clinical outcome? It's not meant to cure the primary disease, but does it address any of the symptoms of the primary disease that result from the nutritional deficiency and or any comorbidities that, again, are secondary to the primary disease, but acting through nutrition? And then again, this question of whether it's a public health or clinical problem, can we classify clinical subgroups with special nutrient requirements? Can you identify them? What is the heterogeneity that's in there around nutritional requirements for any given disease or disease subtype? This again is just to emphasize that many diseases are tissue specific. Some of these diseases are isolated to a particular organ. Some of them are systemic, some of them are both. We have to understand that. It's especially tricky for some organs like the brain that is really isolated from the rest of the body in terms of the exchange of nutrients and exchange of metabolites, and we'll be hearing more about that later by Martha Field. Well, what we do know in terms of the underlying biology, and this has been known for a long time, this is really inspired by Victor Herbert, who thought a lot about these sorts of problems. Victor Herbert contributed much to the field of nutrition, and this area is among them. But we know that disease-related etiology, processes like inflammation, in combination with genetic predisposition, autoimmunity, mitochondrial dysfunction, pharmaceuticals, and trauma, all of these factors that relate to disease, in fact, can influence physiological processes related to nutrition, absorption, transport across the brain or nerve barriers, catabolism of nutrients, excretion of nutrients, altered metabolism, altered tissue distribution of nutrients among tissues. And these are going to impact human nutrition in terms of whole body deficiencies, tissue-specific deficiencies. It could, in turn, give us conditionally essential nutrients or new nutrient toxicities. And it's going to impact our biomarkers, both function and status. We need to begin to think about tissue-specific biomarkers of nutrient deficiency using agents such as cerebral spinal fluid to be able to look at nutritional status. And we're going to need predictive biomarkers in terms of the effect of depleted nutrients in disease and progression of that disease or comorbidities related to that disease. So just some examples of factors that affect nutrient status and or biomarkers of disease. If we think about catabolism, we can think about acquired arginine deficiency syndrome, where the enzyme arginase that degrades arginine is elevated in a variety of different disease states and states of trauma. When that happens, arginine levels are depleted, and often these patients show clinical improvement when arginine is then restored. Now, that's one clinical outcome. Is this a real physiological process? For some reason, why is arginase elevated in disease? Is there some benefit to that? Is there risk to that? We need to understand that. Inflammation is associated with catabolism of certain nutrients. We can talk about tissue redistribution or excretion of nutrients. That is infection. When one shows infection, iron tends to become depleted from the plasma and then is distributed into tissues. Well, does that create a tissue-specific deficiency in other organs? How does that work? And is this beneficial or is this deleterious, depending on what outcome we're looking at? Same thing with redistribution in terms of vitamin B6 and vitamin D. And of course, in disease, we can have decreased rates of uptake across barriers, whether it's the gut or the blood-brain barrier. In terms of stem cells, we have to think about, again, tissue regeneration in disease. This is a figure out of a recent review looking at bone marrow-derived stem cells and the effect that vitamin C has on improving both the quality and quantity of those stem cells. And one of the keys to healthy aging, it's believed, is maintain a good, robust population of stem cells that are healthy and can differentiate and restore the tissue. Well, we know there are a number of nutrients and bioactive food components that in fact support neurogenesis. We also know in terms of muscle fibroblasts that serine is a conditionally essential amino acid for that cell type. For most cells, they don't require serine. They can make it from glucose. For myoblast proliferation, again, this has been known for a long time, they have an absolute essential requirement for serine. Do they have a increased serine need then, the whole body, for tissue regeneration? That's one of the questions that we can ask. We're going to see today that we're going to look for examples of other best practices to try to inform how we think about nutrient needs in chronic disease. And we're going to hear a lot about inborn errors in metabolism, where we know that nutrition is compensating for functional deficits caused by genetics. And inborn errors in metabolism offer really genetics as a proof of principle, where nutrition is compensating for a loss of function due to a genetic defect, and that medical foods have been developed to really get around that roadblock of that genetic loss of function. Well, for chronic disease, can we think about nutrition compensating for functional deficits caused by disease? Is this going to be a good model for us to learn from or not? For this slide here, when my wife saw this, she thought it looked like a Jeopardy board and she wanted hemochromatosis for 200. But at any rate, we can think about how the inborn errors in metabolism example relates to our disease example, where we can think about a physiological change, whether it's impaired nutrient transport, where then we have a special nutritional requirement to overcome a barrier that may be energetic or physical. And we have examples in the inborn errors in metabolism around people who have mutations in the folic acid receptor, and they can't transport then folate across the blood-brain barrier. But we also know that there's some people who have autoantibodies against that same receptor in disease that give us, again, the same phenotype of essentially a brain folate deficiency, and we'll hear about that. We can talk about nutrient depletion due to renal excretion, where we have to provide more input of that nutrient rather, and we have an inborn error of metabolism example, and we have an example where a drug has the same effect. We can talk about nutrient toxicity, where you need to provide less of an input. PKU being an example, hemochromatosis again being the disease example. And we can talk about conditionally essential nutrients, where you have to provide a new input, where you can have a defect in fatty acid oxidation, and then carnitine becomes a required nutrient. You can talk about, as we said, trauma and surgery, where arginine can then become a required nutrient. We can also think about this through the Cochrane-Pico approach. And Patsy Brennan talked a little bit about this, comparing a DRI to a special nutrition need. For the population, we're talking about healthy populations that we can break up into subgroups. Well, we're talking about clinical populations. But can we deal with them as a population? Can we classify them by disease? The intervention here is diet, what you can get in the supermarket. Here, the needs may not be able to be met by a diet that you can get in a supermarket. We want to see dose-response relationships for both, but really the outcomes for a DRI is to avoid nutritional deficiency, to support physiological processes and or chronic disease prevention. Here we're talking about avoiding a disease-induced nutrient deficiency, to support compromised physiological processes in disease and or to support tissue regeneration. So it's really dietary management of that disease. So I'd just like to close by saying this is a really important area and recognized as an important area of research. ASN, when it published in 2013 its research agenda, on that list were one, variability in responses to diet. We know that disease is certainly a contributor to that, so there's some good basic science to be done, and translating that into medical management. So we can see this was a high priority area for the American Society for Nutrition. We also see in the National Nutrition Research Roadmap put out by the federal agencies, question one here, topic one, how do we enhance our understanding of the role of nutrition in health promotion and disease prevention and treatment? And number two, how do we enhance our understanding of individual differences in nutritional status and variability in responses to diet, which again, disease plays a role in. So again, this is a front and center issue in terms of future research agendas. So with that, I'd just like to thank my colleagues, Martha Field, Amanda McFarland, Caperto Garza, and Patsy Brennan, who were helpful in developing this presentation. Thank you.